Hello and welcome to this video podcast for human genetics. This is the first of the last few podcasts for the course. Throughout this semester, we've talked about many fundamental facts of genetics. We talked about what DNA was, what it was made of, how new copies of DNA are made, how that DNA codes for genes which ultimately become proteins, how that DNA is inherited from parents to their children, what kinds of mutations can occur to alter those proteins, which can lead to disease. In these last few podcasts, we're going to talk about how we can and how in the past we have applied this knowledge of genetics. And ultimately, it will be up to society and science to decide what we will do with that knowledge. Sometimes when we think of biotechnology or transgenic organisms, which I'll define in a moment, we might think of some kind of image like this, where mice have been genetically engineered so that they are fluorescent green. Now, making a mouse fluorescent green in itself is not a huge breakthrough, but being able to target specific genes to allow them to be expressed in a particular organism, that is a huge breakthrough. And that kind of technology is what results from images like these. So as we get moving now, let's go ahead and define some terms. The first is biotechnology, which is the alteration of cells, DNA, or biochemicals for a specific application. Those applications could be a variety of things, including a clinical application, an environmental application, and others. Next, let's define recombinant DNA. And that is DNA that is formed by combining DNA from different organisms. And the last term for now I want to define is transgenic organisms. And a transgenic organism is an organism with genetic modifications, often with DNA from different species. These three terms we'll use periodically in this podcast and maybe over the next couple podcasts. But the fluorescent mice we saw in the last image, that is an example of a transgenic organism. The mouse that contains this green fluorescent protein obtained from a jellyfish. That green fluorescent protein that was introduced into the mice, that was recombinant DNA. So now I want to spend just a little bit of time talking about why is it possible that we can use biotechnology in this way. That is, why is it that we can take DNA from, say, humans and put it in bacteria, and now you can make human proteins? Why is it that you can take a gene that makes green fluorescent protein from a jellyfish and put it in a mouse and it expresses that gene? It's important to remember that the genetic code, that is, the code in our DNA that tells us or tells our cells which amino acids to make and ultimately which proteins to make, is shared by all of life. If you look at this diagram here, we can see we have our last universal common ancestor, that's what LUCA stands for, way back here. And it doesn't say it on here, but this occurred about 3.5 billion years ago. We know that eukaryotes here evolved about 2 to 2.5 billion years ago. And then we have animals down here, and if we extended that a little bit more, we would know that humans and chimpanzees, we had our last common ancestor about 5 or 6 million years ago. And then about 2 million years ago, some of our more direct ancestors like Homo habilis and Homo erectus evolved. And then what ultimately we would recognize as Homo sapiens, they first arose about 200,000 years ago. Now, we talked all about that in the last podcast, or the last couple podcasts ago, when we talked about human evolution. So you don't need to memorize those dates again. But the reason I'm telling you all that is that it has been a really, really long time, right? We have used the exact same genetic code for billions of years. It's pretty impressive. The way that genes are made, the same 4 bases, the way that they are replicated, the way they are transcribed into RNA, and the way that RNA is made into proteins hasn't changed in over 1 billion years. Because it works. Because we know that regardless of what we're talking about animals, bacteria, archaea, plants, fungi, we know that they all use the exact same genetic code and they make proteins in the exact same way. So we can exchange that DNA and it'll make that protein. Now that's what we can do on the level of science. That doesn't necessarily mean we should do everything we can. That's where some of the biggest debates have happened and will continue to happen, where science and society meet. And that's what we want to talk about in this and the next few podcasts. Now, in this slide, just a part of a figure from your textbook, and it talks about patenting genes. And I think this is kind of the culmination of a lot of genetic technology and what we can do with it, and where we've had to make some decisions about what we should do with things. And so I just want to go over some of these dates here that I think are important. So the idea of patenting different organisms is not a new idea. The first time that this was done, as far as I can tell, is back in 1930, when new plant variants were patented. Now, this plant that was patented, it wasn't the result of humans genetically modifying it in the way we think of it today. It was the result of humans crossing plants and getting various new plant variants that were better at making a certain product or surviving a certain stress. But those patents were first awarded in 1930. Then over the next, say, 50 years, we got a lot better at understanding the genetics of organisms and how to manipulate that genetics. The first patent that went to genetically modify organism, as you can see here, occurred in 1980, and that was a bacterium that was made that could metabolize components of crude oil. And this was really a big step forward because this helps clean up oil spills that happen in our seas or on land. In 1988, not even a decade later, a mouse that could produce a human protein in its milk was made. Having a mouse that can do this in its milk in itself is not as usable, but that went on to lead to a cow or a sheep being able to produce human proteins in their milk. And then you could mass produce that protein in a cow's milk or sheep's milk, and then it could be purified a lot better, or you could just drink the milk. You see 1992, transgenic cotton. So we started now moving into different organisms. It started becoming much more common. And this is just a partial list. There's a lot of others. In the mid-1990s and towards the end of the 1990s, we started sequencing genes. And we started doing it a lot more efficiently. We were doing it before, but it became more efficient. And so companies then got the idea that, well, maybe we could patent a gene. And this was quite controversial, because we all have these genes, but some company was patenting it and could potentially make money off of genes that were in me and in you and everybody. Should that be allowed? Now, in the early 2000s, our ability to do this was so much greater that we couldn't just say we want to patent gene A. A company would have to say we're patenting gene A because it has this specific potential benefit. They had to explain why it would be useful. Prior to this, some companies were just patenting genes they thought might be useful in the hopes that someday it might make them money. And this really led to about 10 years of people really struggling and arguing and fighting over what should be patented as far as genes and DNA is concerned. Now this culminated in about 2009 when the patent for the breast cancer gene, BRCA1, was being challenged in the courts. And you can see here in 2002 that patent on the breast cancer gene was invalidated. They could no longer hold that patent. And this was huge, right? Because this kind of started to set back some of the 10 years of what some argued was unethical. The government considered changes to gene patent laws, and the U.S. Supreme Court in 2013 declared that genes are unpatentable. So we can no longer patent genes. But at the very least, this is a good example at how new technology entered into the system and how we as a society dealt with that as far as the ethics of how we would use that information. And this will come up time and time again as we move forward with our general understanding and use of the human genome.